Good morning, good afternoon, and good evening from wherever you are watching us from. From my side, I'm having a very good afternoon, and I hope you're also having the same good afternoon. I want to take this opportunity to once again welcome you to this class. Today, as I promised, I want now to talk about paper 2 of KCSE 2009. So remember to subscribe to my channel, remember to follow for more videos, so that anytime I post a video, you'll be the first one to be notified. So without wasting time, my name is Delvin Wenani, and let us go to class. Thank you. Let us meet in the classroom. Now, welcome again. This is KCSE paper 2 of 2019. Now, I'll take you through from question 1 to the last question, that is question number 8. Probably we have question number 7 and number 8 as optional. The paper 2 comprises of 2 sections. First section, that is from question 1 to question number 5. They are compulsory questions. And then question number 6, which is a graphical question, is also a compulsory question. And then question number 7 and question number 8, those are optional questions where a candidate is supposed to choose one of either of the 2 questions. Each question comprises of 20 marks in section B. Section A has also a comparison of a comprehensive mark of 40 marks. 40 marks in section A and 40 marks in section B, totaling to 80 marks in paper 2. So, ladies and gentlemen, in 2019 paper 2, this is what happened. Question 1. In an experiment to investigate the effect of sodium chloride on the growth rate in a spinach seedling, seeds were treated with different concentrations of sodium chloride. The results are as recorded in the table. So we can see from the results the concentration of sodium chloride as mole per liter. You can see the trend from 0.00 to 0.18, a trend of around 0.06, an increased trend. Then percentage of spinach seeds which started to grow roots, you can see from 99.98% to 54.0%. Then you have the mean root length, that is in millimeters. We have from 17.7 ranging down to 7.6 millimeters. So let us go to the question. Question number 1, from the results in the table above, explain the effect of increasing the concentration of sodium chloride. What is the effect of increasing sodium chloride? Now we can see from 0.00, the percentage of spinach seeds was 99.98%. But as we go to 0.06%, a mole per liter, the percentage of spinach seeds goes to 98.20. As we move to 0.12 concentration of sodium chloride moles per liter, the percentage of spinach seeds which started to grow roots moves to 92.0%. So from this we can easily conclude that an increased sodium chloride concentration, or whenever we have more sodium and chloride ions, the solute, that is, decreases osmotic potential and therefore makes water potential more negative outside the seed, and therefore the seedling in the surrounding, that is the solution, the surrounding to become hypertonic to the cells up in the seedling. And therefore what happens? The seeds will take in less water by osmosis. In simple, we say the seeds they become dehydrated. They lose water molecules to the surrounding solution, and therefore reducing the growth rate. We can also talk about the enzyme activities are interfered, and also this one brings about the reduced growth rate. Remember, any other words to that effect shall be accepted as long as the aspect of increasing sodium chloride affecting the seedling, affecting the percentage of spinach is brought out. As long as that point is brought up, that whenever we increase the concentration of sodium chloride, we discover that there's a decrease in which the percentage of spinach seeds which grow roots. Next question. Apart from a ruler, state 2 other equipment one will need to determine the rate of growth in the roots. Now we can use the thread. We can also use a marker pen, and also we can use a die. Remember, in growth we are only measuring the length in growth. So once we have marked the initial length, then we can live for a while to measure the length after. With a reason, state one other part of the seedling the student will focus on to determine the effect of sodium chloride on growth. The question is actually asking us for any other part of growth that can be affected by this concentration. And therefore, these are just the shoot tips, that is the apex, which is also what we call a region of cell division. And therefore, it also will help us to determine if the sodium chloride affects the growth. State the likely effect on the seedling of increasing the seedling, increasing the concentration of the sodium chloride to 2.20 moles per liter, increasing up to 2.20 moles per liter. So whenever we increase the concentration, you saw what was happening. So they want us to set the likely effect once we reach 2.20. That's a very huge concentration. So definitely the seedling will be dehydrated, and hence it will wither, or what we call die. Now, question number 2, the table below shows results of blood cell counts per millimeter cubic of blood from a sample of people living at different altitudes. They want us to explain the relationship between the questions that will come. We have the red blood cells count, and then we have white blood cells count. Now you can see, then we have the altitude in meters. You can see from the table, the red blood cells count is moving from 4.8 as we move up the altitude from 750 meters above sea level. There you can see it's moving from 750 meters up to 1,500, then to 2,250, up to 4,500 meters, that's above sea level. Then the red blood cells are increasing as we're increasing the altitude from 4.8, 5.3, 6.7, 7.6, up to 9.82. But white blood cells is constant. So they want us to explain the relationship between, 1, red blood cells count and the altitude. And from what I've already shown you, you can easily say the number of red blood cells count increases with the increase in altitude. As a mark. And to increase the oxygen carrying capacity. Now that one is a reason why the red blood cells count is increasing. Now since oxygen concentration is lower at a higher altitude, that's another reason. So that one amounts to 3 marks at that point. Then white blood cells count and the altitude. Now we notice that the white blood cells count was not changing, isn't it? It was not changing. It was at 0.45 up to the end, from 750 meters above sea level to 4,500 above sea level. So white blood cells count, there was no increase. Why? First of all, we shall say that white blood cells serve to protect the body against harmful microorganisms, that is pathogens. The quantities of pathogens or vulnerability of the body to microbial attack is not dependent on the altitude. Hence, the number of white blood cells count is constant at whatever length or whatever distance. Explain why chances of nosebleeding increases with altitude in humans. Why there's chance of nosebleeding with increase in altitude in humans. Atmospheric pressure will definitely decrease with the increase in altitude. And this one will create what we call an imbalance between the outer atmospheric pressure at higher altitude and the internal blood pressure, which is generated by the heart. So this results in, 1, nosebleeding when moving to higher altitudes, because the internal blood pressure is higher than the outer atmospheric pressure. Now, state one importance of irritability to living organisms. Now, irritability is the ability of organism to respond to environmental changes. So it enables living organisms to avoid dangerous, harmful, harsh climate, environmental conditions. But it also enables living organisms to obtain or get access to favorable environmental conditions like light, water, oxygen, etc. Any of the two is correct. Now, in an experiment, students treated seedling as illustrated below. We have the first one. This light is coming from a unidirectional source of light. Then you have the first one. We can show, and this one is an affected tip. This is actually a growing shoot. This one is an affected tip. This is a growing tip. Then we have a decapitated tip. Then you have tip covered by an opaque cap. Now, the light is coming from a unidirectional source. You can see from here. Light is coming from a unidirectional source. And therefore, you can see the first one has shown some effect by a growth curvature. The second one has nothing. Then the third one is also showing nothing. So let us go to the question. They're saying, so let us go to the question. Account for the observation made in seedling 1. Account for the observation made in seedling 1. Now, we can see the growth curvature towards the unidirectional light. So it is a positive phototropism, that is a tip bends towards light. Now, this causes the migration of auxins produced at the tip to the darker side of the shoot, resulting in a faster division of cells or elongation on the darker opposite sides of the shoot, and hence bending towards light. We are known of auxins. They are negatively phototropic, that the auxins. And therefore, anytime a light comes from one source, the auxin hormones tend to migrate to the darker side. If the light is coming from one side and hitting a shoot, the auxin hormones tend to move to the darker region. And as they move, they cause an elongation, that is a growth curvature, on that side that it has going to be faster than on this side. And therefore, the shoot will always bend towards the light. Now, explain the similarity in the end results made in seedlings 2 and 3. 2 and 3. Now, both seedlings remain upright, and you can see in seedling 2 does not have the tip, while in seedling 3, the tip has been covered by an opaque material, preventing light from causing an equal distribution of auxins, and therefore there's no growth difference. State the likely treatment that will make seedling 2 and 3 respond like seedling 1. How can you make this is like modification? How can you modify seedling 1 and 3 to respond like seedling 2 and 3 to respond like seedling 1? And therefore, fitting an agar block treated with auxins at the decapitated end of the seedling. 2. Removing the opaque material covering the tip of the seedling. 3. Now and then replacing the opaque cap with a transparent one. So we can do that. In cats, question number 4. In cats, the gene for fur color is sex-linked. Letter G represents the gene for ginger fur color, while letter B represents the gene for black fur color in a given coat species. These genes are codominant. Now, what that means, heterozygous females have ginger and black patches of fur, and heterozygous females have ginger and black patches of fur, and their phenotype is described as tortoise shell. So let us go to the question. Now, with the reference to the information given above, what is meant by the term codominance? called dominant. Now, this is a situation where both alleles are expressed or they show equal distribution in the phenotype of the offspring. So, and therefore, we say none suppresses the other. So, and we are saying the gene for ginger and black fur color will definitely express themselves equally in the phenotype of the offspring. That's why we have the patches in the or their offsprings. Explain why male cats with a tortoise shell phenotype do not usually occur. We talk about once it's a sex. The gene for coat color is sex-linked, and therefore it's contained in the X chromosome. But males have only, or they inherit only one X chromosome. And then they have Y chromosome, which does not carry genes. A tortoise shell female was crossed with a black male. Determine the genotypes and phenotypes of the offspring. A tortoise shell female was crossed with a black male. Determine the genotypes and phenotypes of the offspring. Now, this is from my drawing. That is my drawing. I can explain. Now, the parental phenotype, we have the tortoise shell fur crossed against the black fur male. Then you have the parental genotypes. These are the parental genotypes, and these are the black fur genotypes. Then you have the gametes. These are the gametes. The crossing is done here. The F1, we have the female black fur, and then the female tortoise shell, the male black fur, and the male ginger fur. This is from the tortoise, from the cross between a tortoise shell female and a black male. Now, a person accidentally touches a hot pan and responds as illustrated in the diagram below. That is the hand touching a hot pan, and therefore the forearm is withdrawn. You can see the hand moving up. Explain how the response illustrated above occurs. Explain how the response illustrated above occurs. And I'll be explaining step by step from what occurs. It starts by the stimulus. The stimulus is actually the heat. So once heat is detected by what we call the pain receptors or heat receptors in the skin, which is in this case at the finger of the person, the impulse shall be transmitted along a sensory neuron to the CNS, that is the central nervous system. Now, the transmission will occur across a synapses to a relay neuron. Explain how auxins are utilized as selective weed killers in agriculture. Explain how auxins are utilized as selective weed killers in agriculture. Selective weed killers contain auxins which are absorbed by the weeds, making the weeds to grow abnormally faster and die out ahead of the desired plants. Section B. Now, section B is 40 marks. Now, we're supposed to answer question 6, which is compulsory, and either 7 or 8. As I said, question 6 is a graphical question, so it's a compulsory question. But question 7 and 8, those are optional questions. We can pick either of them, either of the two. So let us go to question number 6. The table below shows the rate of growth of formation. The table below shows the rate of product formation for two enzymes H and J over a range of pH values. So they have the pH value ranging like that. Then you have the rate of product formation for enzyme H, that's milligram per hour, goes up to 4. pH. The rate of production for formation for enzyme J starts from 4, pH 4 up to pH 10. So let us go to the question. On the same axis, plot graphs of the rate of product formation against pH. We are plotting graph of the rate of product formation against pH. So pH will be on the x-axis. Then the rate of product formation will be on the y-axis. So we are going to have two graphs, one showing the rate of enzyme formation for enzymes H, and then the other one showing the rate of product formation for enzyme J. All of them will be in the same axis, but pH will be on the y-axis. So if you do that, you're supposed to obtain something like that, where now we have our pH increasing on this side, and then we have the rate of formation going on this side, the rate of product formation. Now plotting, you'll be given 2 marks for each graph. There are 2 of them. Scale, that is 2 marks. Smoothness of the curves, 2 marks. And then labeling, and you're supposed to label each of the graph. Like this one, you're supposed to label going downwards, and you're not supposed to use a line, just label downwards like this to show that this is a certain one and this one is the other one. So you're supposed to label them. Labeling, you get 2 marks, a totaling of 8 marks. Now, account for the rate of product formation for enzymes H between, now I believe you have plotted them. Now let us respond to the question. pH 1 and pH 3. pH 1 and pH 3. The rate of product formation increases with the increase in pH up to the what we call optimum, then decreases. pH 2.0 is the optimum pH value for the activity of enzymes 2. Between pH 1 and 2, the enzyme molecules are activated. pH 3 and 7, the rate of product formation sharply decreases with the increase in pH. Now, the enzyme is not favored by the increase in pH, and hence it's denatured. So no product forms above or beyond pH 4 to pH 7, where all the enzyme molecules will have been denatured. From the graph, determine the pH value at which the rate of product formation of the 2 enzymes was the same. So we are supposed to see where the 2 graphs are meeting. So where they are meeting, you will read the pH at that point. For this case, I think you can see from your graphs, it is at pH 4. The value of the rate of product formation for enzymes H and I are the pH value stated in C1 above. In C1 above, it is 15.0 milligrams per hour. So at that point where they are meeting, you can read the ranges, that is 15.0 milligrams per hour. The optimum pH value for enzyme J, it is 7.0 pH. Suggest with a reason the likely part of the human alimentary canal where enzymes H will be found. And this is the stomach or any acidic medium, where we say in any acidic medium there's low pH. There's low pH. Question number 7, giving examples, describe the following interaction among organisms. The first one is predator-prey relationship. Then we have symbiosis. Then we have parasitism. Question number 8, explain the effects of increased physical activity on the following organ systems: that the heart, lungs, kidney, and skin. The heart, lungs, kidney, and skin. So ladies and gentlemen, that one brings us to the end of that one. For the essay question, that is question number 7 and question number 8, I'll post all the answers in the comment section. So when you go to my description, you'll find the answers for question number 7 and question number 8.